This is the first video in two different parts where we're going to discuss IB Section 3.5, Genetic Modification and Biotechnology. And this is a really cool, interesting subject that really gets into kind of some forefront topics in biology and really also some possibilities of career fields that if you're interested in continuing studying in biology in college and beyond, you can really get into some cool stuff and lots of new technologies available with biotechnology and different things that we can do now with new technologies that have been invented in the last decade and 15 years or so. The first thing I wanted to discuss is a process called gel electrophoresis. And this is a process that uses a gel. It's kind of like a thicker or more firm type of jello. It's called usually agarose gel. And the process or the purpose of this process, gel electrophoresis, is to separate charged molecules in an electric field according to their size and their shape. And so we can actually do this using DNA, because DNA is a charged particle. And we can extract DNA from eukaryotes or prokaryotes from different cells. And they can be moved through the gel based off of their size and in using an electrical current. And so what we see happen is actually the DNA moves differently depending on how big it is based off of its size. And so small fragments are going to move faster through this gel than larger ones. And so the smaller ones actually move further. So taking a look at our image here, the green is representing the gel. And the DNA samples are loaded into these two wells or two lanes that we have in the gel. And some electric current is applied and the DNA is going to move in this direction as the red arrow, and they're separated by size. And they move from the negative electrode towards the positive electrode because DNA is slightly negative, so it's attracted to that positive electrode, so it moves in this direction. Dye that is added to the DNA binds to it, and then we can actually, once that gel is done running, we can take that and we can look at it under UV light or some different light sources, and we can look at the DNA to see how it's separated. And what we find based off of this is that because of the size, the DNA separates. And this size is based on the amount of BP, and that stands for base pairs. So it's based off of the number of base pairs in that DNA. And what we can use this for is to compare different species to one another. We can use it to compare different individuals within the same species. We can actually even use it to determine genotype, the alleles that are present for specific traits. There's a wide variety of uses for this. And if you do any sort of advanced biology classes, potentially in high school or definitely in college, you'll definitely have an opportunity to practice doing this technique because it's one that's actually used in the lab. And so here's an image of what we would really expect to see from a gel electrophoresis. The fragments of the DNA move in an electric field and they're separated according to their size, moving towards the positive end. These different bands here, if you will, are showing how far the DNA has actually moved. And so these would each be different lanes, different samples, and they've moved different distances. And so on the left-hand side here, this is what we usually call a ladder. And this is used to help us measure how far the bands have traveled through this gel. And so then we can actually visualize them to see which lane, which subject here has moved further. And it can be used, gel electrophoresis can be used in something called DNA profiling, which we'll take a look at in this video here in just a few minutes. Now you may be thinking, well, how do we get enough DNA to actually be able to do this? Because DNA is very small, and it would take a lot of that very small molecule to be able to see it in a gel. And generally how we're able to do this, and most often how we're able to amplify the amount of DNA, is through a process called polymerase chain reaction, or PCR for short. And this process was developed in the 1980s, 1985, by an individual named Kary Mullis. And what this process does is it takes a specific section or small amount of the DNA, specific sequence, and it multiplies that specific sequence many times. And so then what we get is a large amount of a specific section of DNA, and that could be a specific gene or part of a gene, whatever it may be. That amplified DNA then, in using gel electrophoresis, can be used in DNA profiling, we've now been able to use it to identify species, the heredity of diseases, cloning, sequencing, phylogeny, comparing different species, looking at taxonomy, and also looking at parental or in crime scene investigations. All of this is possible, this whole process is possible, by the importance of using something called Taq polymerase. And if you remember back to our DNA units and videos, we talked about DNA polymerase. Taq polymerase is a type of polymerase that was extracted or removed from Taq bacteria. And it's not susceptible to being broken down or to being denatured at a high degree of heat, usually about 94 to 96 degrees. And so that temperature is needed to break apart the DNA, to separate the two DNA strands. And then because this Taq polymerase is able to withstand that high temperature, it's then able to make copies. And so pretty much what's happening in this process is multiple copies are being made of this section of DNA. And so in the PCR process, it's amplifying that DNA, so we get lots of copies of it. The steps to actually conducting and doing PCR, the process uses something called a thermal cycler. And the first part of this is to denature or to separate the two DNA strands to break them apart. The next step is called annealing, and in this step the temperature is decreased to allow the primers, which identify the specific region of DNA that's going to be amplified. By decreasing the temperature, it allows the primers to form hydrogen bonds with the target sequence. The third step is called extension, and this is when the Taq polymerase is adding nucleotides to the three end of the primer, and then those 3 cycles are repeated multiple times, usually about 20 to 40 times. And so you think about it, if you start with one single copy of DNA, after this cycle has gone through once, you now have 2. If the cycle is repeated itself again for a second time, starting with 2, there's now 4. And so if that process repeats itself 20 to 40 times, you're ending with thousands and thousands of copies of the same sequence of DNA. It's like forcing a portion of the DNA to replicate many, many times in a small area. And so with this information, with this amplified DNA, and by using gel electrophoresis, we can do something called DNA profiling. And DNA profiling can be used to identify suspects from trace DNA evidence. If you've ever watched any crime shows, this is what they're doing in those crime shows when they're analyzing DNA evidence. But it can also be used to eliminate innocence from investigations, as well as be used to determine paternity between, trying to determine who the father is of a child between 2 different individuals. And this is all possible because the DNA of each person is unique. It's different between different people. We can also make up something called a genetic profile, and we can use something called genetic markers to compare an individual's set of genetic information, their DNA, to one another. We can also use blood stains or hair samples or semen samples. Basically any body tissue from an individual can then be, DNA can be extracted from those and amplified using PCR in order to be able to conduct this process. And generally the region of the DNA, the part of the DNA that's looked at, is something called a short tandem repeats. And these are repeating, highly repetitive non-coding sequences of DNA that are unique to each individual. And so what we can use these for is a way to create a fingerprint, if you will, a DNA fingerprint. Each person has a unique fingerprint on their hands, while each person also has a unique DNA fingerprint. And by looking at these specific regions of the DNA, we can distinguish between individuals and develop a DNA fingerprint. So let's take a look at an example here. Let's say we have 3 individuals, and we are trying to determine how these individuals compare for their short tandem repeats. If DNA is extracted from these 3 individuals and we're looking at this specific region of DNA, after PCR and after gel electrophoresis, we've conducted that process in order to separate the DNA. We can see that each one of these individuals has bands at different locations on this gel. And so what this is really telling us in this very short, simplified example is that each of these individuals has some different combination of DNA. They're not all exactly the same. There's no similarities here between these 3. In another example, here's a more realistic image. This would represent, this column here would represent DNA found at the crime scene, and then we have 3 different suspects. If you look closely at this, you can probably identify one suspect that has DNA that's matching. And so what this could be used for is to rule out two of the other suspects that would not be linked to this. And so if you take a second to look at it, you've probably noticed that suspect number 2 has the same banding pattern as the DNA found at the crime scene. And so you could then logically conclude that the suspect number 2 is probably at least involved in this crime. Here's another example with the victim and different suspects. And so to wrap up our discussion today about biotechnology is looking at something called the Human Genome Project. And you've probably heard this before. It was a project that took place during the 90s and into the early 2000s. It was coordinated by the U.S. Department of Energy and National Institutes of Health, as well as multiple private and international companies contributed to this. And the goal was to identify all 20 to 25,000 genes in human DNA. So it was to take human DNA samples and to sequence all of the nucleotides in the DNA sequence. This information has been stored in databases that actually you can access, and improves the tools for data analysis for DNA sequences. And from this information, we've been able to identify different types of genetic diseases, classify organisms based on their DNA. We're able to start producing drugs and medications based on an individual gene sequence or protein structure, and genetic diseases also repeated.